Hello! Welcome to the Microbiology Journal Club. This is our microbiology news segment where we talk about all things great that are small. I'm Faz Alam. This is my co-host Danny Chan. We're both microbiologists with who've got some experience, and we're really passionately interested in the science of things and bring that discussion out in the open. And this is what we're doing. So our goal is to talk about scientific papers and discuss the ins and outs of them. But this week we're going to be doing our first look of a bunch of different papers that we will select for perhaps going in deeper next week.
Yeah. Yeah, that's been the format that we've been doing in this time. It's kind of nice, right? You can review something, but we started this journal club really because we want to dive into the figures and sort of absorb like the more detailed nuance points of looking at the data. But there's so much out there that we do this off week thing where we kind of like look at a bunch of the different ones because yeah, there's like so much research.
Yeah, there is not enough time in the day for us to get everything, but we do want to take a look at as much as we can before we try and pick the right ones that we can really dive into. And this is what we're doing this week. We're going to be looking at we're going to be looking at also some of the kind of news that's been going out on COVID especially, because that's the big microbiology story at the moment.
Right. And so we're on Twitter. If you want to follow along and tweet us questions, you can do so using the hashtag or at us. And even between these sessions, we're open to hearing suggestions from people. And we also have a Zotero, which is like a reference manager, shared library where I also add all the stuff that we talked about, and people can see things that we've talked about in the past there as well.
Yeah, and
that's in the doobly-doo.
Yeah, the doobly-doo. Yep, that's YouTube slang.
Okay.
Yeah, we may be stuffy academics, but we can definitely hang with the kids.
Yeah, so this week, I mean, I think we're going to end up talking about one of the papers that we talked about in the previous week. But, news-wise, there is that news that the Russian Federation released a vaccine, and they haven't. And they're explicitly have not done a phase 3 trial.
Yeah. So this is opening up to our usual section that we've added, the COVID vaccine Olympics. Because apparently the way people are treating it is like a game now, rather than people thinking about people's lives who are at stake, and that has been no more apparent than with this news story about Russia, who have now approved a vaccine. Now theoretically, I mean, they say that they're the first ones to approve it, but I mean, I think technically we might talk about this like previously that in China the CanSino vaccine, which is based on adenovirus 5, has been approved to be used in military. So
military,
yeah. But
so that's like I mean, this happens. I feel like this happens with people who are in service because the military is like just a branch of like their bodies are the government's. So like let's use the military as a testing bed, right? Like there's a bunch of healthy people here. We can test in this population. It's kind of weird, but yeah, that's sort of what happened. That's what happened in that instance.
Yeah, it's one of those things that happens quite a lot. And in this Russian news story, it's kind of interesting because, I mean, I've been looking at the news around it, and there are things that, like, the lead scientists have all, like, dosed themselves with the vaccine as well. And and so you keep hearing about, like, and I think I've heard, like, Vladimir Putin has given it to his daughters already, so I mean, there's
one of his daughters.
I mean, I didn't look into the story that deep. I mean,
I mean, it's just bizarre, right? Like, I think that, like, as you said, like, there's a weird game feel to some of this, right? Weird. Where it's like, first to make a vaccine, like, don't worry, guys, like, we got this covered. Like, it's coming for you, so don't get worried.
Yeah, I mean, it's sort of like it's understandable because of the way that the science is communicated. Once people say, "Oh, this trial has been a success," that can mean different things, and I am kind of concerned that going ahead too fast with these vaccines can lead to some safety issues, and because vaccines aren't always safe. I mean, the most notorious one, the most notorious story is the polio vaccine that wasn't killed properly in like the 50s and led to loads of deaths because the vaccine wasn't really safe.
And that's like one for like polio vaccine. They've been making that vaccine for a long time, so that's like one formulation, right,
Yeah.
of the polio vaccine.
So,
So I think that that's important to remember too. That like just if one gets passed, right,
mm,
like everything has to be tested, right? Because where it's made, like how it's made, all of those things impact its safety. Yeah. Well, so there were tests for this Russian one. It's just like phase 1, phase 2 testing, right? They just haven't done the large scale phase 3 testing.
yes.
Um, and when you think about these phase 1 and phase 2s, like I found on their website they link to the clinicaltrials.gov sites. That's the American registry, and it says first group of 9 volunteers, and then second group of 9 volunteers. The second stage will include 20 volunteers. So they're very small.
Yeah.
These are very small groups that they've been tested in so far. And then all this hearsay of like maybe people who are involved with the study dose themselves or like right, but that's not the same as like selecting some broad spectrum of the population, right, to receive the drug.
Yeah, I mean the best thing that basically the only thing that they can really speak to them is their faith that this vaccine won't like cause a massive side effect, which
Right.
I mean, I mean again like for other vaccine candidates like I know I keep trashing Inovio, but I would have a lot more faith if I saw the people like don't electrocute themselves with their own vaccine, but um,
Yeah, you don't want to be electrocuted. I think that this is your fear of being
yeah, I feel that this is my specific bias in that instance. So I mean, it could work, and that's my fear. That my fear is the Inovio vaccine being the most effective vaccine ever. But um, but yeah, no, this is a very very small phase 1 study, and yeah,
And then
so
this is just the registry that we're looking at.
yeah,
It's not even the published thing saying that, after they looked at all of this is what happened.
yeah,
Um,
and again, it's very difficult for. I mean, firstly, it's very small. Secondly, the actual like we don't know whether it works. I mean, we've looked actually in depth at like a couple of vaccines, I think, on this, uh,
mhm.
and like, well, some of them show some promise. I mean, in like animals, in human studies, like we've got literally zero data on what these vaccines will do if people are infected. Um,
Mhm. Mhm.
but there's something I did want to talk about with this because the actual vaccine candidate is quite interesting though. Um,
Mm.
So we've talked about like the adenovirus so the Chadox vaccine, which is based on adenovirus. I forgot which one it was. Uh, chimpanzee
Chimpanzee
adenovirus,
adenovirus. I don't
yes.
know. Yeah.
And there's like the CanSino, which is based on human adenovirus 5,
Hmm.
and there's another vaccine by Janssen, I think is adenovirus 26. Um,
Mm.
so these are all species of human adenovirus, and one of the things that has come out in the CanSino studies is that there is a concern that humans who've already had these adenoviruses might be already immune to them. So the moment this
Hmm.
vaccine goes there, it doesn't manage to enter any cells to cause any immunity because they get cleared out by the immune system. Um,
Hmm.
so, and also the concern is that you can't give boosters because adenoviruses are so readily cleared out by the immune system. Once you give it once, that's like the immunity to the adenovirus will be there. So the second one boost won't have the same
Hmm.
effect. But one of the
Hmm.
ways that this vaccine gets around that is by using two different vaccines. So they both present the same antigen, but one of them is on an adenovirus 26 and the other one is on adenovirus 5. So your booster is so you don't have to worry about so you've got that initial booster and you've got a second booster and you don't have to worry about that additional like adenovirus immunity. I mean,
Yeah. Yeah. Trying to get the same. Yeah. That idea of adenovirus at the end of the day here is it's a delivery mechanism, right? And
right.
you want to be able to deliver this construct into cells so they can make the antigen. And the way it is interesting to think the way they're getting around is just vary the specific species, right, of adenovirus or the strain of the adenovirus.
Yeah, and so this is almost based on a previous vaccine that this institute created, which was for Ebola.
Mm.
So I did look at that, and the Ebola so the Ebola vaccine they took that to phase 1 and phase 2 trials, and So what they did in that one was they used a vesicular stomatitis virus, a VSV virus, and
Hmm.
and an adenovirus. So the first one was the VSV virus that was a primer, and the second one was the adenovirus 5. Um, but so I mean,
So again, just another virus as a delivery
yeah.
mechanism.
So that's the thing that I'd be interested when they actually publish their data, because this is different because they said that this vaccine, the reason why they were so fast, because this vaccine is very similar to what they've done in the past with this Ebola vaccine,
Hmm.
but they changed out the entire virus, and the question is,
Sure.
what was the reason for that? I mean, if they had this virus ready to go that could express their protein, why doesn't change? Especially when there is always that concern with the human adenoviruses, that there is already going to be immunity, that some level of the population isn't going to be affected by them.
Hmm.
So yeah,
Yeah, I mean, lots of unanswered questions, I think. I think that's the thing. And that's why, I think the international, right, scientific community has also been like, this is just kind of strange and suspect, right? Like, we shouldn't be endorsing types of actions like this because
yeah,
it skirts a lot of the regular safety protocols that we'd see.
yeah. I mean,
But again, like at the end of the day, like it gets tested in humans. So, this is why I sort of like think about how testing on the military seems kind of okay. Their bodies like belong to the state in a way that like regular citizens don't. And then here it's just like, I think the consent, to me, it's like a consent issue,
mm,
even. Like, you could think of it like that. Like, okay, so they have some initial idea here. Can you really consent people and say, like, are you willing to take on the risk of taking this when like you don't know the risks of taking this object? Um, it just changes that, right? Like it changes that idea of like what is healthcare, right? Because people who could take this early, they're not really getting something that has that much data behind it. In some ways, they are becoming the data,
yeah,
right? Because they're going to be the first. They're going to be the ones that are getting tested. And like again, like that's similar to this idea of testing in the military. It's like you have to get it in some amount of bodies before you can say that it's safe, right? So they go to this like pool of people that they have. Yeah, but like for everyday citizens, I feel like that's I don't know. That's like too much of a leap to ask of people.
yeah. I mean, although there are people out there who are willing to like play guinea pigs. I mean, I think there's like maybe 10,000 people sign up to challenge studies or like volunteer themselves to be infected, which is
Yes.
like
Yeah.
crazy pants to. I mean, I understand why
Yeah.
because
I actually think it's a UK website. It's
yeah,
like it's something about like one I don't remember what it was, but it's like it's a bunch of people, volunteers, and you sign up and like if a challenge study happens, they have a pool of volunteers ready to go.
yeah, it's interesting. Because again, I think there are some dosing studies at the moment to figure out what the right dose of COVID to give people for challenge studies. I'm not sure. I haven't heard much more about that. Um,
Hmm.
and I know that
Yeah,
yeah,
I pulled one of the papers. I think it's like a white paper, but it was studied in Science. Uh, oh, maybe it's called A Strategic Approach to the COVID-19 Vaccine. Corey et al. Let's see. Let me open this. I might have. Yeah, yeah, yeah. Um,
a strategic approach to the COVID-19. So
yeah,
by Corey, and I know Anthony Fauci, who's like
yeah.
a celebrity, is like listed on that. Um,
Right. But basically, it talks a lot about different vaccine things, and in it they do mention the human challenge studies and how that might be a way of that might be a way of doing these. Mainly, I think it's like because with vaccines you have to wait for someone to get infected, and then so
hmm,
if you give someone a vaccine, they're not necessarily going to be infected down the line. So the idea is that you infect them, and that makes the sample size that you have to use smaller. But I think something to remember about this as well, if you're doing human challenge studies, is that people are chosen to be challenged based on their profile that they aren't likely to have severe disease
hmm,
from this. And so you're already seeing, right? You're going to see some sort of bias in there as well, in the type of efficacy that's coming out, right? Like it's
yeah,
effective in this population that's being screened in this way. And not to say that that's like a nail in the coffin, like this shouldn't happen. But it's one of those things where it's like there's gradations to it, right? Oh, it sounds really good. All these healthy people can volunteer and help us get this vaccine faster. But on the other hand, like that introduces a new bias into the data. That's like, right, like those vaccines that get approved in this way may not have the broad type of efficacy that we want, like in the elderly, or who are like
Yeah.
a really important population to be able to vaccinate.
Yeah, I mean it's a tough challenge to think about because I mean it really depends on what you're going for. Because if you're going for like trying to stop severe cases like pneumonia and other sorts of things like that, then you do want to focus on the elderly. But if you want to look at sterilizing immunity and to cut down the transmission cycle, then you are going to have a vaccine that produces sterilizing immunity in healthy people, like who form the majority of the people who will carry the disease, but
Sure.
can. So that's the tough thing. That's the one thing that we can't really test for properly at the moment, not with the phase
Hmm.
one or phase two, because all we can get is antibody titers. We can get proxies for how well like this we might be able to fight the disease, but we don't know, and it's going to be
Hmm.
very hard to
Hmm.
get that date because you can rush like phase one and phase two trials kind of because that's where you're looking for safety. Phase three you can't rush at all because you have to wait for enough people to get infected, and that can take some
Hmm.
time. And also to get in order to get
Hmm.
outcomes, and odds are when the data comes in, it might not look how you want it to look. And so if you're going,
Hmm.
so if you like looking like in very short term, you can end up like either screening out candidates that could be good or letting you through candidates that are bad. So that will have to take time. So the idea of having a vaccine that's actually ready and proven by say October or November is very very unlikely. I'd be
Yeah.
very
Yeah.
surprised if that were the case. But I guess that's why,
It's
yeah,
I think it's interesting because I feel like there's definitely been all this media being like, oh, like people are compressing the timelines, it's moving faster than ever, right? But I also see, I think I sense in like the periphery now, it's more like, oh, but realistically, like how long are we going to wait? Don't get your hopes up too much. Like things do take time,
mm,
and like having a lot of public pressure to have something faster, like it probably encourages, right, this type of action, right,
yeah.
of jumping the gun a little bit. So it's good to also like, yeah, maybe take a step back and be like, okay, well, what should we be doing until then, right? And those things are still important, right? Like, what are we doing about testing, right? Because like that's what we have going forward, right?
Yeah,
Like at this point right now, that's all we have.
yeah, exactly. And there,
Which actually, oh,
no, no,
I
you,
just want to say that that segued interesting into I found a preprint that talks about where is it? Oh yeah, test sensitivity is secondary to frequency and turnaround time for COVID-19 surveillance.
to
So this is
frequent.
a preprint, and it was published a while back. Um, but I don't know. I've been talking about testing more, and this came up. It's kind of it's interesting, right? Because we talk about like how frequently should we be testing people, and should like the accuracy of the test is really important, right, to get more
Mm,
tests out there. And then this paper kind of is trying to argue for maybe less accurate tests, but that can be deployed more often,
so,
as a way of yeah, monitoring outbreaks.
so this is Larry Morris, all I presume, like, because I've knocked it up on the screen. So the idea that,
Yep.
yeah,
Mm-hmm.
so
Yep. This is the one. Learn more.
yeah, I mean, yeah, it's interesting because I guess if you got a test with a certain like so if you have a test that isn't perfect but it gets it results
Mhm.
some of the time, just doing multiple amounts of that test can actually cover that kind of issue.
Yeah, yeah, absolutely. I think that's what they're getting at. They're trying to make an argument for it at least. And I think it's interesting
Hmm.
because there were when like maybe last week in New York City, I think because the states was the rising cases in the states, like getting tested, people waited like almost 2 weeks to get their
Mm.
results back. And at that point, it's not useful, right? Because like you could have just quarantined yourself for those 2 weeks if you thought that you had it.
Yeah.
So like that test doesn't play a role, like testing doesn't play a role of like surveilling yourself because you're only finding out after the fact, after you've done 2 weeks of activity where you might have had a positive. It doesn't help you like modify your behavior. And I think in this one, it's saying that it could. But I think also I think there's assumptions here too. Like, what do people do with their test data, right? Like, just because you're getting tested, like, doesn't mean you're going to necessarily change your behavior.
Yeah, I mean that's something that people will be studying on for ages afterwards. I mean, I've seen like there are some like modeling studies that take mobile phone data to like has a proxy for how much mobility and how effective lockdowns are. Those tend to be the most pessimistic models. So
Yeah, well, because like phones go, yeah, I think it just shows how much activity is really happening, right?
yeah,
Like people are like running around and stuff.
yeah.
Yeah,
So
so anyways, that's something about testing. I also on the in the testing line, this test Saliva Direct,
oh
simple and sensitive molecular diagnostic test for SARS-CoV-2 surveillance.
right, I think I've heard about that.
Also a preprint came out earlier this year, and we haven't talked about any diagnostics, so this might be a time to talk about them. Um, and
Yeah,
yeah, basically, they're trying a protocol of saliva tests.
yeah. I mean that's interesting because I mean because the thing I'm very interested in at the moment is whether there's going to be a T cell test, but at the moment I haven't seen anything like that because
Oh
it
yeah,
because it
is
go
that just because we read those T cell papers?
yeah, there's that T cell
Yeah.
paper that had
Mhm.
where they had the control group that was tested via the antibody test. Except we know that the antibody tests aren't necessarily as reliable as we'd like, so there's no way to know whether that whether the author's
Right,
assertion that was a cold virus that caused
right,
that caused the crossover was actually true or not. But
right,
that
right.
actually brings me to one of the papers I wanted to suggest.
Oh
So
yeah, please.
because one way to get around that is to check blood samples from people that were taken before the outbreak. So there's this
Hmm.
one paper that was published in. Let me see whether I can find it quickly. Let me uh, switch back. So yeah, because the problem with because I think I highlighted the fact that the other paper they looked at people, but the only way they selected for them was whether they had the antibody test, and the
Yeah.
people in the control group had some protection from COVID, and so
Yeah.
the big question
No,
was
not wait, was it protection? It was that
sorry.
some of their T cells recognized, right? They they came negative on all these tests for COVID, being that they didn't have any active infection and they didn't have antibodies to it, but their T cells still came up recognizing specifically, right, the antigens from SARS-CoV-2.
Yeah, so I've got selective and cross-reactive SARS-CoV-2 T cell epitopes in unexposed humans.
Yeah.
Um, So this study looks at it's very similar to the T cell receptor study we looked at before, but except it finds a similar effect in blood samples taken from 2018 and 2019, which are such halcyon years, back when we were in the outdoors and we didn't know what coronavirus was. So
Oh,
so
this is another study about T cells.
yeah, so I mean
I see.
we've covered that, but I think that this kind of
Yes.
strengthens the author's assertions in their study, in the previous study, that that is due to
Hmm.
like pre-existing cross-reactivity with other like viruses.
Hmm.
So
Hmm.
I find that quite interesting. Um, yeah, well, uh, and yeah,
Yeah,
so
that's
that,
interesting.
but that's why I've been quite interested in looking at like, so saliva direct. I think that's been something that because I know that I've seen a couple of websites that are quite impressed by it, and
I mean, of course they want to impress you. I mean, because like at the end of the day, like this is like the market at play, right? All these vaccines, all these diagnostic tests, right? Everyone is vying for, yeah, a spot to sell, and I mean, hopefully do good, right, through
yeah,
distributing these tests. But yeah, there's definitely something flashy about it.
yeah, I mean, I think there is something to this because I know like kind of when I was testing vaccines and things like that, that saliva is a really good kind of proxy for. Firstly, it's very easy to get, but I mean, taking blood
Yeah,
samples is it needs a lot of training and needs a lot of like kind of. But anybody can give themselves a saliva test, so that's actually
right.
something that's why this is a lot more because I've seen lots of diagnostic papers come out with these funny tests, but this is the first one
Hmm.
that actually like looked kind of like it's something that we can actually apply quite easily as like normal people?
Yeah, yeah. I mean, it's interesting. I think that there's many ways to do the test. I got swabbed for a COVID-19 test recently. It does take a bit of like it's not something you
Hmm.
could do to yourself, or even that your family members could do to you, because it goes so
Oh
deep
yeah,
back into your nasopharynx.
yeah.
But
That's like
and then my sister had one. You can do it through the throat too, like as long as you get back there somehow. But it's like a very long stick that has to go up into the back.
yeah, man. Let me think.
Uh,
So there has been some more.
so I
Sorry,
mean,
you go for
I
it.
was just gonna say, uh, I mean, this is sort of related. We're talking about antibodies. I think this is something that was in our docket before, but again, like part of knowing what to test for in antibody tests, right, is knowing like what antibody responses look like in people who have been infected. Um, and so I think like we're looking at it, but I just want to say that, you know, this is an extension again of when we were talking about that T cell stuff.
Hmm.
Like what does it look like in infected people, right?
Yeah.
Like trying to dig in, understand what that looks like, because I mean it helps for antibody design, but I guess where we're talking now is a bit on the testing side, right? Like what would you even test for, right, in terms of antibodies with people who are infected?
Hmm. I've got some more vaccine papers to talk about because
Yeah,
we've
sure.
finally got some data from the killed cell vaccine. So
The what?
killed
Kill.
so the inactivated vaccine. So
Mm.
this is from not Sinovac, the other Sinopharm. Um, so let's see if I. So yeah, I've got the effect of inactivated SARS-CoV-2 on safety and immunogenicity outcomes. Um, so this is an interesting one. Well, again, this is another phase 1/2 trial. Um, but except it, well, actually no, it's weird because they've actually like done the phase 1 and 2 trials. So they've basically looked at the first like clinical trial, and they've also looking at the second one all in one like go. So in the first one they've got like 96
Hmm.
randomized, but like again 24 per like group, which basically amounts to about 12 people getting the vaccine, and then the second one is a bit bigger, where it's randomized with like 84 or so 84 28 84 28. Um, so it's a bit of a bigger study, so there's more chance of picking up more rarer side effects. Um, and again, they've got like looking at various antibody responses.
Hmm.
I don't know if this is showing up. Okay, um, so looking at the antibody responses in the phase 1 and in the phase 2 trial. So they've got like
Hmm.
uh, and they've got like quite a lot of data, but it's yeah. So but I mean, it seems okay. It's not really much more exciting than what we've seen previously. Uh,
Yeah, I mean it's another one, but I mean this is the clinical data that goes along with, because previously we'd only seen the mouse data,
yeah,
right, on this. So
yeah. So this one is, and this is the one vaccine that has been we haven't really heard much about it, but this is the most traditional vaccine of them all. The
hmm.
the the original
Hmm.
recipe of how you create a vaccine is you take the virus, you, I guess kill is the wrong word, but inactivate it using various
Yeah.
chemicals, and then inject that back into patients using like an adjuvant, and then you can,
Hmm.
and that's a technique that's been shown to work in multiple occasions. Um,
Oh
the only
yeah.
yeah
I mean again, it's one of the classic ways of doing things.
yeah I mean, the only issue that with this might be the side effect that people are really looking for is a potential for like the immune response to be made accidentally more severe, because sometimes viruses have ways of playing tricks with the immune system to make the immune system more severe or more like dangerous. So let me see what I can pull up that. So
Yeah, is that like the dengue, or like the classic example is dengue fever
yeah.
there, right? With like antibody mediated entry, something like this. So the antibodies that get produced like also help the virus enter cells better.
So yeah, antibody dependent enhancement of SARS-CoV-2.
Hmm.
That's what people are worried about. And so the reason,
Hmm.
I've pulled up a study from 2016 which looks at immunodominant SARS coronavirus epitopes in humans elicited in enhancing and neutralizing effects on infection in non-human primates. So basically, they took some. So by the looks of it, they found that's that certain epitopes that attack like the SARS coronavirus can actually make the disease worse through antibody means.
And this is with classic SARS, right? Is
Yeah,
what
this
they
was
did.
classic old school SARS. So this isn't new SARS. In fact, I did look around. There are plenty of people like who are afraid of this and have managed to like write articles, but no actual data on this yet. So I mean, this could be like jumping at shadows, but we want to be very careful because SARS-CoV-2 and CoV-1 they are related, and if they have the same effect, then we want to be aware of that because we definitely do not want to have this kind of effect happening in SARS-CoV. We don't want
Hmm.
to elicit this in our vaccinations. So the worst case scenario is if you have a vaccination that actually makes the disease worse. So whenever people are looking at like studies and vaccine safety, one of the things they're looking for, in addition to like the basic allergic reactions, cytokine storms, are this is one of the things that people are going to be looking out for
Hmm.
in later trials. And
Hmm.
one of the reasons why like rushing isn't a great idea. Let's see what I mean. I say rush. I didn't say Russian. Rushing. Just wants to
Rushing the vaccine.
rushing the vaccine. Um,
Accelerating past the regular process.
yeah. Let's see. There's another like study that caught my eye, which is uh, so single intranasal dose of chimpanzee adenovirus vectored vaccine confers sterilizing immunity against SARS-CoV-2 infection. Now, this study is interesting because
that that's not Chadox.
it's not Chadox. No, this they use
Oh,
a
another one.
different. They use adenovirus 36 for their. I think they use a different chimpanzee adenovirus, and
Sure.
but still based on the S protein, spike protein. I just recognized one of the authors, and uh, yeah, yeah, it's this is interesting because it's a mouse study, and we did spend some length last week talking about the ins and outs and why mouse studies may not be the most beneficial thing. But the interesting
Hmm.
thing I found out about this is that they induce sterilizing immunity, but the reason they did that was because they used a different way of applying the vaccine. So when they applied the vaccine intravenously, they didn't get the sterilizing immunity. They got the immunity that lessened the systemic infection, but when
Yep.
they applied it intranasally, that produced sterilizing immunity, which
Hmm.
I find quite interesting because the study we looked at with the Chadox vaccine which was looking at chimpanzee, well not chimpanzees, but looking at rhesus macaques. They were applying the
Yeah.
vaccine intramuscularly, and
Yes.
so and they've
But
changed
with this
that.
different, it's like in this different model. It has like the way of delivery really changes
Well,
the
I
immunity
mean,
that comes.
yeah, because I think there's like the idea of like different immune compartments. So like there's systemic immunity and then there's mucosal immunity. So the immunity of like kind of your respiratory surfaces. And so the
Hmm.
interesting thing here is if you have a vaccine that is applied to those surfaces, and then that could so like you can get an immunity in the area where you want, so where the vaccine where the
Yeah.
virus first hits. Um, and the thing that
And the flu virus, the flu vaccine comes in that the flu vaccine comes that way as well.
yeah, so that's
There
what
are injection versions, and there's also ones that you snort.
yeah, and that's what got me kind of interested in this because I feel that this is why I'm going to pay attention to this because it's not something people generally think about. People often think about the chemistry of vaccines, but actually how you deliver them can be very important. So this is
Hmm.
why I was quite interested in this study because that's the main like thrust of it. That it's not what you give, it's how you give it. Um,
Sure.
but yeah, And
And so
well,
they do in this paper they cover both routes of administration.
yeah, they do. So they looked at they looked at like intramuscular dosing as well as like intranasal dosing, and they found that they like so the intramuscular dosing induced a robust like immune response, but a single intranasal dose also did the same thing, and also completely prevented infection in both the upper and lower respiratory tracts. So
It's interesting.
that is interesting. And the interesting thing is, I think the Chaddock study are also experimenting with intranasal dosing as well. So that might be an interest. So I hope if there's a difference there, then that'd be an interesting avenue to look at. Um, so what other things did I find? Cold adapted live attenuated SARS-CoV-2 vaccine. So again, this is another mouse study,
Oh,
but they
that
use
that that is the way that the flu intranasal one is delivered.
yeah. So the full title is Cold Adapted Live Attenuated SARS-CoV-2 Vaccine Completely Protects Human ACE2 Transgenic Mice from SARS-CoV-2 Infection. So these are so as we discussed last week. So ACE2 transgenic mice can actually get a SARS-CoV-2 infection. So this mouse model probably has a lot more kind of value than the one we talked in the previous study. And the cold adapted live attenuated. This is again old school vaccinology, where so again that's something that is somewhat interesting. Let's see what else. And the
Well,
last
the idea is that cold attenuated, right? Because is it old school? I'm not sure. I know it for the flu. It's that idea that it can't infect deeper in your body because it
yeah.
needs the temperature inhibits it.
Yeah, so yeah, exactly. So it gets used to surviving cold temperatures, so the human body's temperature can completely annihilate it. So, I mean, there's good and bad sides to live attenuated vaccines because if you evolve a virus to be attenuated, then it can always evolve back if you don't look after your vaccine stocks properly. So
Hmm.
it's something that I thought found interesting, and also Novavax. Novavax have been developing a nanoparticle vaccine. So it's made out of the recombinant spike protein. So if I recall correctly, I think this is, uh, yeah. So I think it's bound, but it's got multiple RBDs, or no? It is just like a trimeric version of the spike glycoprotein. So I think it's like a three version, and it's a nanoparticle, and so they've got
Yeah.
like
So this is like when we looked at the BNT1 or whatever, the
yeah,
Pfizer. One of the Pfizer candidates was a nanoparticle based one as well, but they're ultimately not moving forward with that one.
yeah.
So yeah,
I
here's
mean
another.
yeah, the thing about the Pfizer one, it was an mRNA vaccine, so it forced your body to create the nanoparticles. Uh, whereas this one,
Oh,
like,
this they're making it.
they're actually making it and delivering it. Um,
Hmm.
so again, this is another phase one trial with maybe some added like antigen studies, and so I feel like calling things phase one slash two really uh, can misrepresent it because I don't think any of the studies we looked at that were phase one slash two were. Um, oh yeah, do you want the link to this? The first in-human trial of a SARS-CoV-2 recombinant spike protein nanoparticle vaccine uh, by Cheryl
Hmm.
Keach et al. So uh, this one is, I mean, I guess it looks promising because I mean, some people, I mean, there's been good people saying good things about all the vaccines. Um, But yeah, so this is yet another like I mean at least say the phase one sorry I was going on a rant about the phase one slash two idea because the phase two trials the
Hmm.
whole thing that makes them valuable is that you testing in more people rather than like getting
Hmm.
that first like hint of whether like the new trait whether there's neutralizing antibodies or not. So a lot of the phase one trials, one slash two trials we've been looking at have been on very small numbers of like between 10 to 12 people. Um, so
Yeah,
giving them
under
that
20
moniker
for
of
sure.
phase two, I feel is maybe giving a bit too much hope. Not that hope's a bad thing, but um, yeah,
But it's like we talked about this. Like it's also just because for investors and stuff, right? You
yeah,
want to see that things are moving along well. So, but it does like So this is like decidedly just a phase one.
Yes, this.
Is that what they're saying?
Yeah, they're
Interesting.
saying it's just at phase one. I mean, I think they're being quite honest about that because they are still taking a lot of the data that we've seen in the phase 1/2 trials, where they do take
Hmm.
they take out like T cells and test them whether they're antigen specific, and they look at the antibody
Sure.
responses. So I feel like yeah, this is, and they say first in human. I don't think they say phase 1/2, or they don't seem to be messing around like
Yeah.
that. But yeah, so it
Well, I mean, it's just a different set of buzzwords,
yeah,
right?
half of science is diving through the buzzwords. Uh,
Mhm.
um, yeah, let's see what other things
Hmm.
that I've. Oh, right. So have you been hearing about camelid antibodies and the new spray that they've developed?
No, but I mean, I know
Oh,
chemical antibodies, nanobodies.
nanobodies. Yeah. So so I've been seeing like kind of it lighting up on Twitter how this like there's this potential for a spray that can kill off like COVID or stop it from infecting based on nanobodies. And so
Sure.
they've basically like
Very
got this.
small. This is what's nanobot.
Yeah,
It's exactly what it sounds like, right? Small antibodies. It's actually
yeah,
just it's like camels just have antibodies that are a bit smaller, but they the region that the antigen recognizing region is smaller, so easier to make, I think is one of the ideas because antibodies are actually quite large proteins.
yeah. So I've got two like studies on nanobodies. Like so, one of them was looking at like an ultra like high affinity synthetic nanobody blocks SARS-CoV-2 infection by locking spike into an inactive conformation. Very descriptive title.
Hmm.
And the idea, and they basically like in this one they've developed a synthetic nanobody that they say is very stable, stable enough for you to aerosolize it and to like deliver it to
I
patients.
think that's the other advantage of nanobodies. People have always said, right? They're
Yeah,
smaller. They don't have the same protease sites that, like, yeah,
yeah.
monoclonals do.
I mean they're very stable, and I mean it's interesting to look at kind of how these antibodies work because, and also they're also quite good to. I think they're relatively inexpensive to produce. I guess maybe I'm not sure, but
Well, they're shorter, right?
yeah,
So shorter proteins are easier to produce.
yeah. And I guess the stability, the fact that you've got less protein to have to worry about, means that you can actually make it more stable. So this study, they look at like the stability and whether it can be aerosolized. There isn't much. I don't think there's any treatment data on it. So it mostly like talk. So I did a brief once over it, and it doesn't look there's any testing in like any live models or anything like that. But I mean, it's interesting nonetheless, and it's one of those things that has been getting some oxygen in the press. Um, yeah, so because I know we've heard a lot of like about so the nanobodies are interesting, but at the moment so this is a story I'll be keeping an eye on because if there's treatment data out there, that'll be really a good thing to look at, and it's an interesting idea of just spraying
Sure.
antibodies into the air. Um,
It's like yeah, I mean certainly like that's a lot different than therapeutic
hmm,
monoclonals, which we saw in the mouse model paper. I don't know why this one doesn't have a DOI, but when I was searching for the first nanobody one, I also came across neutralizing nanobodies bind SARS-CoV-2 spike RBD and block interaction with ACE2.
yes. So let me see if I can.
Because nature hides their DOIs at the bottom, that's why.
Ah, so let me.
Okay. So do you want to talk about a nanoparticle vaccine, right?
I mean,
Do you want to talk about another vaccine paper
oh, so
necessarily?
yeah, I feel like I'm not sure about that yet. I do want to like talk about something that's in human studies.
Like the first in human trial.
So the first in human trial of I mean, of the inactivated. I mean, the thing is, there isn't much to dig into with that. It is the
Right.
only thing that we can really look at is like the increase in numbers. But I mean, if there was one of the ones I would like to look into, it'd be the CanSino one, because of that. kind of big side effect of the part of the population already being unable to have that, and seeing how they explore that problem. I feel like that's kind of that make because while might make the vaccine less like kind of useful, it makes this paper much more interesting to look at, and because I guess it's one of the
That one
things
is which paper is that one?
so I think that's on the Zotero page. Let me see if I can find it.
Oh,
Um,
we just
under
know
vaccines.
we just looked at it today. You're saying the one that was published about CanSino, right?
Yeah,
That's
the one that was published. So we discussed that about 2 weeks ago in the previous. So that would be. So I mean, so we just did discuss that briefly because I feel like since there are a couple of.
Yeah, it's the JAMO one, but then we
Yeah,
also have the one where they did it in mice,
yeah. I'm quite
showing
interested.
the infective
Yeah.
inactivated vaccine against SARS-CoV-2.
Yeah.
And then what else do I have here? Oh yeah, okay. I found again, I think I really looked at older ones. Some of these papers aren't new, but I just think they're interesting. Comparative replication and immune activation profiles of SARS-CoV-2 and SARS-CoV in human lungs and ex vivo study.
Okay.
So ex vivo human lung tissue infected with the virus.
Okay. Uh,
And I'm not sure if that one's more interesting than previously in the animal models folder. We have one where they made a organoid culture and they infected that with SARS-CoV-2.
okay. I mean, I'm quite interested in immunogenicity and safety of the recombinant adenovirus 5 vector by Zhu et al., which is their Lancet study that looks into the recombinant vaccine that has that problem and they with the previous immunity. Um,
Hmm.
so that's of all the vaccine studies, that's the one that's most interesting because to me, like having a vaccine study that has a flaw is quite interesting to me. Um,
ZHU, ZHU.
ZHU. Yeah, I'm sorry, I'm not really great at the pronunciation.
Oh no, I just want to make sure that I'm doing it right. It's just hard
Yeah,
always to know.
yeah. Um, but there are other things that we can
Ah,
be
this
looking at.
one.
Yeah. So, I mean, the nanobody is good if we want to look into like the kind of protein interaction, I guess. Um, but I think the adenovirus 5 is something
They
that's
also use the word first in human in the zoo. That's what I'm saying. First in human is just another way of saying phase one.
yeah,
That has like a little bit more of a ring to it. Recombinant adenovirus type 5. So is this the same? So
so
this is so yeah, this is an adenovirus one. Yeah, not inactivated. You're interested in seeing adenovirus. I see. What you're interested in is the how does immunity impact adenovirus vectors if they
yeah,
do?
uh, yeah, that's
That's like the comparison with. It'd be really nice to see the comparison with Chadox or chimpanzee. I almost wonder whether or not if that's the question, we should look at it at some point. But there will be other studies about that aren't with SARS, right? Because it doesn't matter that they're delivering SARS-CoV-2,
hmm,
right? Like that's very much like tell me about human responses to these vectors, and like these vectors are all previously studied for the most part, I think.
right.
I feel like if that's the major question, I mean that's an option for papers that we could look for next time.
Mm. Yeah, I mean, I just think it's kind of like
uh,
yeah, okay. Let's see what else do we have.
I
So
also picked up, oh, there's like there's a paper about SARS-CoV-2 like evolution again, evolutionary origins of SARS-CoV-2, lineage responsible for the COVID-19 pandemic. It's a
yeah,
Nature Microbiology.
okay,
Boney et al.
okay. A minute. I think I've got that up. Let's see if I can.
If you have the Zotero up, my selections are put in in the
Oh,
potentials
yeah. Okay.
folder.
Uh,
That's where I've been adding the ones that we've talked about on this as well.
so yeah. I mean, it's interesting because I've been hearing a bit about the about like kind of the because there has been this idea going around that it was a manufactured virus, and it would be
Oh my
interesting.
gosh.
Yeah.
Terrible. I mean, TWIV like regular this week in virology regularly has to respond to viewer letters being like this is not the case, or like someone will say like I someone told me they heard news about this. I think it like crops up on Twitter periodically.
Oh yeah. No, I saw this like really like kind of big like think piece about it and about the whole like kind of directed evolution, like the again like the like back in like 2011 when they were working on those ferret studies where they had like
Hmm.
d- uh, had was it dual use research was the term where they had like these uh,
Yeah,
and the idea that it'd be very difficult to pick up, and even if you do pick them up, it's going to be quite because again like it's very difficult to pick out the difference between like something that's been passaged in say a lab and something that got accidentally passaged on a farm because
for
those are
sure.
two very equal, and so been a lot of like back and forth about that. So yeah, I'm not too. I mean, it could be an interesting thing to look into. Uh,
And then also have there is another T-cell paper. I don't know. This one's like yeah, this was weird. I feel like maybe we didn't talk about this when we did our T-cell paper,
mm,
but there's a targets of T-cell responses to SARS-CoV-2 coronavirus in humans with COVID-19 disease
okay.
uh, and unexposed individuals. I don't know. It came out in May, but it's another one we're looking at T-cells. Maybe that's too much T-cells now.
Let's see what else is there. There's let's see. I mean, SARS-CoV-2 cell entry depends on ACE2 and TMPRSS2 is blocked by.
Oh yeah, there's also this. Yeah, we really didn't talk about this little section of things.
Yeah.
Um, yeah,
So
because also this is like the structure design. There's also this is structure design, right? That you can look at, like getting closer to um, the biochemistry, right, of the two proteins binding. And then one of the ways people design drugs is just like try every shape that might block that, right? Every small molecule that might block that.
yeah. So I mean, because I think one thing were looking for was some better idea of like the SARS-CoV-2 pathogenesis. This is like what
Yeah,
on the molecular
absolutely.
level. So like
Yep.
exactly what is happening when, and
Yeah.
this isn't exactly a new one, but I feel like this is something that we've kind of neglected a little bit because again, like I do tend to have a bias against like cell based research, even though this was from a treatment perspective, but from molecular biology perspective, it's actually really useful and interesting.
Yeah, absolutely. I like this one because I feel like this is also like a really important one. This is the paper that got cited a lot, just talking about when people were talking about the ACE2 receptor being the receptor.
Yeah,
Right? This is the one where they're saying this is ACE2, and it gives us like another protein as well.
yeah. I mean, I did see some things about furin cleavage, but again, it was
Hmm.
in I saw most of them in bioarchive, and I didn't really. Again, my bias, I was like not very interested in compared to because I found like a bunch of
Yeah,
vaccine papers, so I was like, meh. But
yeah,
this might
yeah.
be
No, fear and cleavage is also important, I think, and it may come into the story as well if we read for the background of this paper.
so. Yeah,
Um,
let's
yeah,
see if
that's
I can.
pre-processing of the spike protein, I think. Fear and
Yeah.
cleavage.
Uh, so let me see if I.
Yeah, I'd be down for this one.
Yeah, okay, I'd be down for this. Looking into the molecular biology of how SARS-CoV-2
Yeah,
causes
yeah,
infection.
because again, it's like it's something that we haven't quite talked about yet.
Yeah, uh, I think that this is. Yeah, I think I'd be happy to choose this one. So
Mhm.
that'd be SARS-CoV-2 cell entry depends on ACE2 and TMPRSS2 and is blocked by clinically proven protease inhibitor. So
Mhm.
yeah, I think that from So I'm not sure about this from a treatment perspective, but from an actual like understanding how this
Yeah,
virus
yeah. I think
works.
that's the important thing. It's not exactly telling us like something that could be used in humans, but it's telling us a lot about the molecular biology of
Yeah,
the binding. Hmm.
like in a petri dish, like seaweed extract is better than a lot of protease inhibitors. I mean, it's so. I mean, it could, hopefully that can be turned into treatment. But again, I'm still skeptical that most things can work in a petri dish that don't really work in real situations. So, but what a
Right,
protease
right.
inhibitor can tell you is like what is being blocked and how the virus is working by breaking something in the virus. So,
Yeah.
and yeah, and a lot of microbiology research is by chemically breaking small processes to figure out how the bacteria works. So,
Yes, yes, yes.
so this is like some really proper like kind of uh, basic research microbiology that they've
Hmm.
spun to make it sound like it could be treatment, but no, this is like proper like
Right,
understanding.
right, right.
Yeah, no, I think
Yeah.
that we've come to a decision. So hopefully you'll join us. Hopefully the audience members will join us next week so we can delve deep into this and we can understand a bit more about SARS-CoV-2 pathogenesis.
Hmm.
Yeah, I mean molecular
Sounds good.
pathogenesis because pathogenesis is a big word. So I
Yes.
yeah.
Sounds
But
good.
yeah, sounds good. Thank you so much for joining me. We'll chat next week.